Alright, next we're going to look at Chapter 3, Part B. So we'll look at active transport. There are two types of active processes, active transport and vesicular transport. And there's primary and secondary active transport, and vesicular transport has three types. All of these types are going to require the use of ATP, which is adenosine triphosphate. This energy source is going to be able to move solutes across a plasma membrane against their concentration gradients. And just as the term down its concentration gradient was a key for passive transport, something moving against its concentration gradient is going to be a key phrase for active transport. Any solute that is too large for a channel or carrier protein, that is hydrophilic or not lipid soluble, or anything that just needs help moving with not down its concentration gradient, not with its concentration gradient, but against it, then that's going to use active transport. This also requires carrier proteins. We would call these carrier proteins that use ATP solute pumps. So just like facilitated diffusion, but with energy. Again, move solutes against concentration gradient. That's going to be the key for active transport. The types of active transport, so we'll look at that first before we look at vesicular transport, are primary and secondary active transport. Primary active transport is pretty simple. We're going to move things from an area of low concentration gradient all the way to an area of high concentration gradient using ATP, or the breakdown of ATP, which is called ATP hydrolysis, to power this process. The sodium-potassium pump is commonly used during active transport. We'll talk about it later on when we look at the nervous system, and we'll mention it briefly when we talk about skeletal muscle. But it's located in all plasma membranes, so it's a really efficient pump and it's readily available. It's involved in primary and secondary active transport of nutrients and ions. And we're looking at it because it is the most studied pump. It's one of the first pumps that functions or works in active transport that was studied. And it's essentially an enzyme. So it's an enzyme that uses energy from the breakdown of ATP in order to pump out 3 sodium molecules from a cell and then bring in 2 potassium molecules. And these are charged atoms, so they're essentially ions. Essentially located in all plasma membranes. It's very easy and useful to study, but it's also active in really excitable cells like those of the nervous system and muscles, which are able to move the body or act as a communicating system. And even though we'll talk about electrochemical gradients later on, these types of pumps are helpful in maintaining that electrochemical gradient. So here are the basic steps of movement for sodium and potassium ions through the sodium-potassium pump. Because it sometimes functions as an enzyme or is considered an enzyme, this pump is also sometimes referred to as the sodium-potassium ATPase. ACE is going to be a suffix commonly associated with enzymes. And again, we're going to break down ATP in order to move and change the shape of this carrier protein, which again acting as a pump. And only a pump because it's using energy. So we're going to start at step 1, where 3 cytoplasmic sodium ions bind to the pump protein. They can only bind to the pump protein after the pump protein has bound ATP. Right after the 3 cytoplasmic sodium bind to the pump protein, the ATP will be broken down into adenosine diphosphate, or ADP, and inorganic phosphate. That breakdown causes a shape change, and so we will have the pump go from facing inside the cell to facing outside the cell. At that point, it will release its sodium, so it'll release the 3 cytoplasmic sodium out to the extracellular environment. And what will be left of the broken down ATP is a phosphate bound to the pump. When the phosphate is removed after 2 potassium from the extracellular environment have bound to the pump, that's when we'll get a shape change that brings those 2 potassium into the cell, because that shape change will open the pump back up to the inside of the cell, and that way the potassium can be released. Then the process gets to start all over again, because the inorganic phosphate leaving leaves space for a new ATP to bind, and then we start all over again. So if I were to simplify that, what I would say is 3 sodium bind from inside the cell, ATP is bound to the cell. When ATP breaks down, the protein pump changes shape and releases the sodium to outside the cell. Then the pump binds to potassium. The phosphate that's left after the breakdown of ATP pops off, and then there's a shape change where the 2-potassium can now enter the cell. When the phosphate's gone, new ATP can bind, and the process starts all over again. It's generally described in 6 steps, and the breakdown of ATP is ATP hydrolysis, and the addition of a phosphate to the protein is going to be phosphorylation. The removal of the phosphate is dephosphorylation. Here's another way to look at it. So this is, instead of being a circle or a cycle, which is what this really is, it's just the steps listed again in 6. So cytoplasmic sodium binds, there's 3 that bind. When ATP breaks down through the shape change, the 3 sodium will leave the cell. 2 potassium bind, the phosphate comes off. Shape change, potassium enter the cell, and then the process will start all over again with a new ATP binds. So active transport uses the sodium-potassium ATPase, but there are also leakage channels located in membranes that result in leaking of sodium and potassium. And so even though the sodium ATPase is the main active transport protein and it's moving sodium, it's moving potassium, some of it's also leaking in and out of the cell. So the sodium-potassium pump is also having to work as an antiporter, which is something that moves 2 molecules in opposite directions, and we were moving sodium out and potassium into the cell. And we need this antiporter to maintain the electrochemical gradients, because if you notice, sodium and potassium are both charged. And so moving those charged molecules in different directions changes the charges inside and outside of a cell locally. And this is essential for functions of muscle and nerve tissues in their cells. So while primary active transport uses the sodium ATPase to move things like sodium and potassium against their concentration gradient, so moving them from areas of low concentration to high concentration, secondary active transport doesn't directly use ATP, but uses the energy generated from primary active transport to move something else. I've already introduced the term antiport or antiporter, but antiport systems transport one substance into a cell while transporting another substance out of the cell, so they transport things in opposite directions. There's also symporter systems that transport 2 substances in the same direction, so either 2 substances in or 2 substances out. Now, secondary active transport can be either one of these, but it doesn't directly require ATP, but it does require the energy from primary active transport having occurred prior. So the required energy for secondary active transport is obtained indirectly from the use of ATP, because indirectly there will be ionic gradients that were created during primary active transport by using ATP. So let's just look at an example of that. So on the left we have primary active transport, and you can see that we're moving sodium out of the cell. Even though there's already a lot of sodium outside the cell, we're moving sodium from an area of low to an area of high concentration. And there is some potassium out here, but we're moving it from an area of low concentration to an area of high concentration. Because even though it's not shown, there would be more potassium inside the cell. So that's going to be a gradient that's established later on when we get to skeletal muscle, but sodium and potassium are moving against their concentration gradients. They're moving from low to high concentration. And this is all possible because ATP is being used. There's an antiporter function that's being allowed because of the use directly of molecular energy. Whereas secondary active transport is going to use the energy created from moving these ions, which used ATP indirectly, to move more stuff. So what's going to happen is we have these kind of like leakage channels, and as sodium moves into the cell, so from extracellular to intracellular, the movement of this charged molecule, which the movement was only possible because we pumped so many sodium out of the cell, the movement of this charged particle is going to pull something else with it, in this case, glucose. So what we see here with the secondary active transport is the generation of a high gradient of sodium outside the cell is able to move glucose into the cell when these leakage channels act as symporters and allow some of that sodium back in. So as the sodium diffuses back across the membrane through a membrane cotransporter protein, it drives glucose against its concentration gradient into the cell. Now, glucose is being pulled in, but it's not going from high to low concentration. There's not as much glucose outside the cell as there is inside the cell. So we are moving something from a low to high concentration area, and that's why it's technically active transport. Even though we don't directly use ATP, we're just indirectly using it. So if I were to summarize primary active transport and secondary active transport, I would say primary active transport directly uses ATP, while secondary active transport does not. But they both move things from low to high concentrations, and by doing so, they're doing the opposite of passive transport, and they're using some type of energy, whether it's indirectly created by ATP or not. Next, we're going to look at vesicular transport. And this is the transport of even larger substances, so bigger than ions or small simple sugars, things like macromolecules, so small proteins, or even small carbohydrates, or disaccharides, or even small polysaccharides. But we're moving these things or stuff dissolved in fluids across the plasma membrane via these membranous sacs called vesicles. And this requires ATP, so it's a type of active transport. The types of vesicular transport that we're going to look at are endocytosis, exocytosis, transcytosis, and substance vesicular trafficking. Endocytosis is when we bring things into the cell. Exocytosis when we bring things out of the cell. Transcytosis is when we bring things into the cell, across the cell, then out of the cell. And substance vesicular trafficking is when we move things from one organelle to another. So here is transcytosis. If we bring large things, even if they're dissolved in fluid, into the cell, that's endocytosis. As they move across the cell, that's transcytosis. And as things exit, things like waste products, that's exocytosis. Oftentimes when a cell takes things in, it will combine the vesicle with an endosome, and sometimes endosomes combine with lysosomes to digest whatever is in the vesicle. So let's look at 3 types of endocytosis. All 3 of these types of endocytosis are going to be bringing things, large things, into the cell. Penocytosis, you can think of it as cell drinking. We're bringing in extracellular fluid that contains dissolved solute or dissolved molecules. So this is one way for a cell to absorb nutrients that are already slightly broken down. But these substances are usually slightly larger than would be just absorbed through the plasma membrane or with the help of, say, a channel or carrier protein. So penocytosis is a fluid phase endocytosis, and we saw the plasma membrane enfold. Let me go back. It brought in the extracellular fluid with solute dissolved in that fluid. And one example of where this works in the human body is in the small intestine. It really helps us absorb nutrients like monosaccharides, also things like amino acids, just the broke down parts of macromolecules. Then there's phagocytosis, and if penocytosis is cell drinking, then phagocytosis is cell eating. We see that we're taking something in and we're going to encapsulate it, and these parts of the vesicle forming are called pseudopods, and when they completely close, we've created a phagosome. So that's a membrane sac that contains a solid particle. And the phagosome generally also combines with a lysosome, again, to digest it down, because we don't have substances that are dissolved already in fluid like we did with penocytosis. And anything that remains that's waste that is undigestible would be ejected out of the cell via exocytosis. Then you have receptor-mediated endocytosis, which is basically just the cell being picky. It's the cell using receptors to attach itself to something specific that it wants to take in. So the uptake of enzymes or low-density lipoproteins, iron, insulin, things like that would use receptor-mediated endocytosis. So let's just look at all 3 together. Again, they're all endocytosis. Things are coming into the cell. Phagocytosis, cell eating, so we have a solid particle being taken into a phagosome. Penocytosis is cell drinking, so we have the cell taking in dissolved solutes into a vesicle. Then receptor-mediated endocytosis is the cell using receptors to bind to something specific and take it in. Exocytosis is the opposite of endocytosis, and there's not as many types. We're just ejecting things like waste, or maybe secreting things like mucus or saliva. Hormone secretion also would use exocytosis, or even neurotransmitter release. But exocytosis is a vesicular transport process ejecting or allowing things to leave the cell and going from the cell's intracellular environment to the extracellular fluid. So here is a photomicrograph of a cell secreting a substance. So this is an example of exocytosis. And just to summarize our active processes, we had primary and secondary active transport. Secondary active transport indirectly uses ATP but relies on the ion gradient created during primary active transport. Our best example of primary active transport is the sodium-potassium ATPase, which acts as an antiporter to move sodium out of the cell and potassium in. And both with primary and active and secondary active transport, we are seeing things move from a low concentration gradient to a high concentration gradient, which was the opposite of passive transport. Then exocytosis, we had the three types of endocytosis we looked at, which are next, but exocytosis was the ejection of substances, and it could be waste, or it could be things like hormones and neurotransmitters. Then we had the three types of endocytosis, which were phagocytosis, pinocytosis, and receptor-mediated endocytosis. So if you forgot those, make sure to go back and look at that overview slide. But here's a really good summary of the active processes, and examples are included. For the most part, they all use directly the energy source ATP. Alright, next we'll continue looking at the plasma membrane, we'll look at cell-environment interactions, and then go into the different types of organelles and their functions.